Chapter 2: Innate Immunity. The immediate response to infection. Physical barriers colonized by commensal microorganisms protect against infection by pathogens. Let's explore how our body's physical barriers, colonized by commensal microorganisms, protect us from infection by pathogens. This chart highlights 3 layers of protection: mechanical, chemical, and microbiology across 4 key body sites: the skin, gut, lungs, and eyes, nose, and oral cavity. First, mechanical barriers. At all sites, epithelial cells are joined by tight junctions, forming a continuous protective layer. In the skin and gut, there's a longitudinal flow of air or fluid that helps sweep away potential invaders. In the lungs, movement of mucus by cilia traps and expels pathogens. And in the eyes, nose, and oral cavity, tears and nasal cilia help wash out microorganisms. Next, chemical barriers. On the skin, fatty acids and antimicrobial peptides inhibit microbial growth. In the gut, a low pH environment, antimicrobial enzymes and peptides serve as defenses. The lungs utilize pulmonary surfactant and antimicrobial peptides. And in the eyes, nose, and mouth, antimicrobial enzymes in tears and saliva, along with antimicrobial peptides, offer protection. Finally, microbiology barriers. Normal microbiota, the beneficial microorganisms naturally living on and in us, occupy niches and help suppress the growth of pathogens. This is especially crucial in the gut, skin, and lungs. Together, these barriers form the body's first line of defense in the immune system, working in harmony with our commensal microorganisms to keep us healthy. Intracellular and extracellular pathogens require different types of immune response. Our immune system tailors its response depending on where a pathogen is located, outside or inside our cells. On the left, we see extracellular pathogens, which exist outside host cells. These include bacteria, fungi, parasites, and some viruses before they enter cells. The immune system uses mechanisms like antibodies, phagocytes, and complement proteins to detect and eliminate these threats in the extracellular environment. On the right, we shift focus to intracellular pathogens, those that invade and replicate within our cells. This group includes viruses that enter host cells, hijack their machinery, and replicate and assemble inside. Some bacteria and parasites can also survive within host cells. To combat these invaders, the immune system relies on specialized responses such as cytotoxic T cells and intracellular sensors that detect signs of infection from within. The location of the pathogen, extracellular or intracellular, determines how the immune system responds to protect the body. Complement is a system of plasma proteins that mark pathogens for destruction. The complement system is made up of plasma proteins that identify and mark pathogens like bacteria for elimination. At the center of this process is a key protein called C3. When C3 is activated, it undergoes cleavage, producing 2 fragments, C3a and C3b. The larger fragment, C3b, attaches directly to the surface of a bacterium, effectively tagging it for destruction by immune cells. This process is known as opsonization, which enhances the ability of phagocytes to recognize and engulf the pathogen. Meanwhile, C3 plays a different role. It acts as a chemical signal that recruits phagocytes to the site of infection, increasing the local immune response. Together, C3a and C3b coordinate to amplify our body's ability to detect and destroy invaders quickly and efficiently. Let's take a deeper dive into the molecular mechanics of complement activation, specifically how the protein C3 is cleaved and becomes capable of tagging pathogens. It all begins with the cleavage of C3 into two fragments, C3a and C3b. This cleavage exposes a highly reactive thioester bond within the C3b fragment. This exposed thioester is unstable and ready to react. If no pathogen surface is nearby, a water molecule attacks the bond, producing soluble C3b, which quickly loses its ability to bind. However, if a pathogen is close, molecules containing hydroxyl, O or amine, NH groups can react with the thioester. This reaction covalently binds C3b to the pathogen surface, a key step in marking it for destruction. This mechanism allows the immune system to target and label only nearby invaders, making the complement system both powerful and precise. The complement system is a group of plasma proteins that help mark pathogens for destruction. It can be triggered through three pathways. The alternative pathway acts first and is initiated when a pathogen surface creates a suitable environment. The lectin pathway follows, activated by mannose binding lectin binding to the pathogen. Finally, the classical pathway is triggered by antibodies or C-reactive protein binding to pathogen antigens. All pathways converge on the cleavage of C3 into C3a and C3b. C3b binds to the pathogen surface, leading to three critical outcomes: recruitment of inflammatory cells, opsonization that enhances phagocytosis, and direct killing through membrane perforation. Together, these mechanisms result in the death of the pathogen. At the start of an infection, complement activation proceeds by the alternative pathway. At the start of an infection, the alternative pathway is the first to activate. In plasma, C3 spontaneously hydrolyzes to form IgE3, which can bind factor B. Once bound, factor D cleaves factor B, producing IgE3BB, a soluble form of C3 convertase. This enzyme rapidly cleaves more C3 into C3a and C3b. The C3b fragments can then attach to the pathogen surface, marking it for immune attack and propagating the complement cascade. The alternative C3 convertase, made of C3b bound to BB, forms on the pathogen surface. This enzyme plays a key role by cleaving more C3 into C3a and C3b, amplifying the immune response and coating the pathogen for destruction. Once C3b binds to a pathogen surface, it recruits factor B. Factor D cleaves B, forming the C3 convertase C3bBB, which rapidly cleaves more C3 into C3a and C3b. This creates a positive feedback loop, coating the pathogen with C3b and marking it for immune attack. Regulatory proteins determine the extent and site of C3b deposition. To control C3b activity, Properdin stabilizes C3 convertase on pathogen surfaces, enhancing immune response. Meanwhile, factor H and factor I inactivate free C3b by cleaving it into IgE3b. On human cell surfaces, protective proteins like DAF and MCP prevent damage by disrupting C3 convertase, ensuring C3b doesn't attack host cells. Phagocytosis by macrophages provides a first line of cellular defense against invading microorganisms. Complement activation deposits C3b on the surface of invading bacteria. CR1 receptors on macrophages recognize and bind to these C3b tags, marking the bacterium for destruction. The macrophage engulfs the bacterium through endocytosis, forming a vesicle called a phagosome. This phagosome then fuses with lysosomes, forming a phagolysosome, an acidic, enzyme-rich compartment that digests and destroys the bacterium. The terminal complement proteins lyse pathogens by forming membrane pores. Once complement protein C5 is activated, it is cleaved into C5b, which initiates the assembly of the membrane attack complex. C6 binds to and stabilizes C5b, creating a site for the next proteins to attach. C7 then binds, and this interaction exposes a hydrophobic region that allows the complex to anchor to the pathogen's membrane. C8 follows, embedding more deeply into the membrane and beginning the disruption of membrane integrity. Finally, C9 molecules polymerize at the site, forming a membrane-spanning pore. This pore compromises the cell's structure, allowing fluid to rush in and causing the pathogen to swell, burst, and die. This diagram shows how complement protein C5 is activated by the alternative C5 convertase, C3bB, on the surface of a pathogen. C5 approaches the C5 convertase complex, where it is cleaved into two fragments, C5a and C5b. C5a is released and functions as a potent inflammatory mediator, while C5b remains and initiates the assembly of the membrane attack complex, MAC, which ultimately forms a pore in the pathogen's membrane. This pore formation leads to cell lysis and death of the pathogen. Complement protein 5, once activated, produces a fragment that initiates the formation of a membrane attack complex. Complement protein 6 binds to and stabilizes this fragment. Complement protein 7 then attaches and exposes a part of itself that allows it to insert into the membrane of the pathogen. Complement protein 8 joins the complex and begins forming a pore in the membrane. Finally, complement protein 9 molecules gather and assemble into a complete ring-like structure that spans the membrane. This entire structure disrupts the integrity of the pathogen's membrane, leading to cell death. The resulting holes in the membrane can be seen in microscopic images and are referred to as membrane damage. This image shows the step-by-step assembly of a membrane attack complex, which forms a pore in the membrane of a pathogen, leading to its destruction. The process begins when the activated complement protein 5 attaches to the surface of the pathogen. Complement protein 6 then binds to this complex and stabilizes it. Next, complement protein 7 joins and inserts itself into the membrane. After that, complement protein 8 binds and also embeds into the membrane. Finally, multiple molecules of complement protein 9 are recruited and assemble into a ring-shaped pore that spans the membrane. This pore disrupts the integrity of the pathogen's membrane, allowing contents to leak out and ultimately leading to the death of the pathogen. This image shows an electron micrograph of a pathogen's membrane after attack by the membrane attack complex. The dark circular structures labeled as membrane lesions are actual pores formed by the complement system's terminal proteins. These pores penetrate and disrupt the lipid bilayer of the pathogen. As a result, the integrity of the membrane is compromised, leading to leakage of the cell's contents and eventually causing cell lysis and death. This is the final destructive step in the complement cascade. In this image, we see a comparison between how the complement system affects pathogens versus human cells. On the left side, the complement proteins C5, C6, C7, C8, and C9 work together on the surface of a pathogen to form a structure known as the membrane attack complex. This complex pierces the pathogen's membrane by creating a pore, which ultimately compromises the pathogen's integrity and leads to its destruction. On the right side, the scene shifts to a human cell. Here, a protective protein called CD59 intervenes. When the complement proteins C5, C6, C7, and C8 begin to assemble on the human cell surface, CD59 binds to the developing complex. By doing this, it prevents the final protein, C9, from joining the structure. Without C9, the pore cannot form, and the human cell is spared from damage. This delicate balance highlights how the immune system can differentiate between harmful invaders and the body's own cells. Small peptides released during complement activation induce local inflammation. This image tells the story of how small fragments released during complement activation help initiate and amplify local inflammation. It begins with anaphylatoxins, small peptides such as C3a and C5e, which are generated during the complement cascade. These molecules act on nearby blood vessels, triggering them to become more permeable. As vascular permeability increases, it allows fluids and important proteins from the blood, such as complement components and other plasma proteins, to leak into the surrounding infected tissue. The effects don't stop there. These complement fragments also serve as chemical signals that guide immune cells, particularly monocytes and neutrophils, out of the bloodstream and into the affected tissues. Once inside, these immune cells ramp up their ability to destroy microbes. Together, the increase in vascular permeability and the influx of powerful immune cells build a potent inflammatory response at the site of infection, marking the beginning of the body's defense against microbial invasion. Several classes of plasma protein limit the spread of infection. This illustration reveals how a specialized plasma protein called alpha-2 macroglobulin helps limit the spread of infection by trapping harmful microbial enzymes, or proteases. It starts with the alpha-2 macroglobulin lying in wait with a bait region and a reactive thioester bond exposed. When a pathogen secretes a protease, the enzyme is lured into this bait region. The protease, thinking it's about to perform its usual function, cleaves the bait. But instead of continuing on, this action triggers a dramatic conformational change in the structure of the alpha-2 macroglobulin. Like a trap snapping shut, the protein wraps around the protease and holds it tightly. Not only is the protease now physically enclosed, but a covalent bond also forms between the protease and the macroglobulin, locking it in place. With its movement and activity halted, the enzyme can no longer break down host proteins or assist in microbial spread. This mechanism is part of the body's elegant strategy to neutralize invaders and keep infection from spreading. Antimicrobial peptides kill pathogens by perturbing their membranes. This figure shows how antimicrobial peptides, such as human beta-1 defensin, eliminate pathogens by disrupting their membranes. The image on the left reveals the structure of beta-1 defensin, a small protein that folds in a way that allows it to interact with microbial membranes. On the right, the diagram demonstrates how defensins are drawn toward microbial membranes through electrostatic attraction. Because defensins are positively charged, they are attracted to the negatively charged outer surfaces of microbial membranes. A transmembrane electric field helps pull the defensins into the membrane's lipid bilayer. Once embedded, defensin peptides come together to form pores. These pores disturb the membrane's integrity, compromising the pathogen's ability to maintain its internal environment. This disruption causes the microbe to lose essential molecules and ions, ultimately leading to its death. This figure illustrates the location and role of Paneth cells in the intestine, emphasizing their importance in producing antimicrobial peptides, especially defensins. The upper part of the image shows the structure of the intestinal lining, with finger-like projections called villi and inward folds known as crypts. Zooming in on a single crypt, we see that Paneth cells are located at the base, adjacent to intestinal stem cells. Paneth cells are packed with granules and have a prominent rough endoplasmic reticulum, highlighting their role in synthesizing and secreting proteins. These cells are the primary source of defensins in the gut, which they release into the intestinal lumen to combat microbial invaders. By secreting defensins, Paneth cells create a protective chemical barrier that helps maintain the balance between the host and the vast number of microbes present in the intestinal environment. This chart organizes human defenses by class, name, synthesis site, defended tissues, and how their production is regulated. The alpha class includes HNP1 through HNP4, HD5, and HD6. HNP1, HNP2, and HNP3 are synthesized primarily by neutrophils, with other immune cells like monocytes, macrophages, natural killer cells, B cells, and T cells contributing to HNP2. HNP4 is synthesized exclusively by neutrophils. These defensins act on tissues such as the intestinal epithelium, placenta, and cervical mucus plug. While the exact tissue targets of HNP4 are unknown, all four are constitutively produced, meaning they are constantly present without requiring infection to stimulate synthesis. HD5 and HD6, also in the alpha class, are produced by Paneth cells in the intestine. HD5 is additionally produced by vaginal epithelial cells. These defensins defend a broad range of tissues, including the salivary glands, gastrointestinal tract, eyes, female genital tract, and breast milk. Their production is both constitutive and can be upregulated, especially in response to sexually transmitted infections in the case of HD5. The beta class includes HBD1 through HBD4. HBD1 is produced by epithelial cells and some immune cells, including monocytes, macrophages, dendritic cells, and keratinocytes. HBD2 and HBD3 are also synthesized by epithelial cells, with HBD3 additionally involving monocytes and macrophages. HBD4 is produced by epithelial cells in the stomach and testes. These defensins defend a wide variety of tissues, including the gastrointestinal and respiratory tracts, urogenital tract, skin, salivary glands, kidneys, and even blood plasma. Their production is regulated both constitutively and in response to infection. Together, these antimicrobial peptides provide a broad and layered defense across multiple tissues in the body. Pentraxins are plasma proteins of innate immunity that bind microorganisms and target them to phagocytes. Pentraxin proteins are key players in the innate immune response, helping to identify and clear invading microorganisms by targeting them to phagocytes. There are two major types of pentraxins: short pentraxins and long pentraxins. The short pentraxin, known as serum amyloid P component, is produced by liver hepatocytes. It plays a broad defensive role capable of binding to a wide variety of pathogens including bacteria, viruses, fungi, and parasites. In contrast, the long pentraxin PTX3 is synthesized by a more diverse set of cells, including monocytes, macrophages, dendritic cells, endothelial cells, and epithelial cells. PTX3 shares similar ligand specificity with the short pentraxin, but is limited to binding bacteria, viruses, and fungi. Together, these pentraxins act as soluble pattern recognition molecules, bridging the gap between pathogen detection and phagocytic clearance, thus enhancing innate immunity.